Lecture. So today we'll discuss the genetic disorders, which are followed with the pathogenesis, and we'll discuss pathogenesis of these disorders. So it's not only enough to learn every genetic disease, but you have to understand the general principle of progression of these diseases. So that's why we have two lectures. In the first lecture, I'll explain the main principles of these diseases on a few examples, and in second lecture, we'll discuss the manifestation of these diseases and how they are present, and some specific types of diseases will be discussed in details. So now let's talk about genetic and pediatric diseases. Okay. So what is genetic disease? It's always confusion in all over the world is that genetic diseases they're pathology of genes only. So when genes are bad genes we have, yes, or might be so everyone who have bad genes they have genetic disorders. Who have good genes they don't have genetic disorder. So that's actually it's wrong, of course. Every of us, every human of the world, every person of the world, they have some bad genes. Bad genes means that some mutations which happen during the life. So as we discussed in last topics of neoplasia in tumors, we discussed that we have a so you remember we have a lot of genetic diseases of one cell, of personal cell, and of course we have genetic disease of personal cell during the mitotic process. So we have the same genetic disease in not same, I mean, but also in the same way we have a lot of potential genetic disease in an entire body. So because genes are spreading there, and that's a feature you have to keep in mind. So here let's check it out. So okay, this thing that only 25,000 protein coding genes for fewer than that 100,000 previously estimated, and almost half the number in the lowly rice plant. So you see, genes we have only 25,000 genes, but these genes are they don't work individually. So yeah, they don't work individually, and they work as a group of genes, and sometimes one gene splits into different parts. So that's okay. Um, we discussed it in very first lecture, so I don't want to spend the time over this. But don't think that so all the inherited diseases they are genetic diseases. We have a lot of hereditary disorders which are actually present congenital. So they're congenital. So this implies it's present at birth. So these congenital diseases they are not genetic. For example, congenital syphilis, yeah, it's shared by parents, so by mother to child, and it's congenital, but it's not related to genetics. Yeah, so that's why congenital is not similar, not equals to genetic. Okay, not all genetic diseases are congenital. The expression of Huntington disease, for example, begins only after the third or fourth decade of life. So you see a lot of genetic lesions which potentially present in our body. They are silent until during the reproductory period. So okay, we'll encounter these examples, few of these examples, but you already understand that. So evolution worked in this way. Yes, that evolution has evolution works only for ones who survived, and we can imagine that a lot of people who can survive until 40 years, but not more than 40 years, they can spread their genes. They can fertilize and born a lot of offspring. Yeah, and that's why they will survive in a case of genetic in evolutionary race. Yeah, and that's why a lot of genes which we will discuss today, they are not present after the birth. So they are not manifested after the birth. Some of them are manifested after the long time living. So, and okay, that's what we are going to discuss today. So, nature of genetic abnormalities contributing to human disease. So there are mutations in protein coding genes. I remind you that protein coding genes, there's approximately 1% of DNA in our DNA sequence. So 1 to 2%. So it's not a lot. Yeah, and there's mutations which happen there. So which happen there, they directly they change the triplet composition, right? This triplet composition of genes. You remember the sequence of DNA is we can split it into triplets, and these triplets encode definitely amino acid, which will be attached with the help of transporting RNA, and this transport RNA will cause the primary structure of protein. Yeah, so you have to remember it. If we have mutation in this gene, so what might happen? So actually, so let's discuss what might happen. We have point mutations. Point mutation. So it's a result from substitution of a single nucleotide base by a different base, resulting in the replacement of the one amino acid by another in the protein product. For example, so this example, everyone knows this example. So it's mutation in beta globin chain. So it gives rise to sickle cell anemia. Sickle cell anemia. So this is point mutation. So only one nucleotide is replaced, only one. But another amino acid get attached to the beta globin, and the entire shape of the globin changed, and normal size of normal shape of entire cell is changed. And due to this, sickle cell anemia happens. Just 1 point mutation. Okay. Then frame shift mutation. Frame shift mutation, it's when during the deletion or insertion happens. So that's easily, you can easily understand it. So you have a sequence of nucleotides, and then just imagine we have one extra nucleotide, or we delete one nucleotide. So all the frame of triplets is shifted, and then encoded absolutely um, different amino acids. And of course, it's reading frame of the DNA strand is shifted, and of course, no proper proteins are produced. Of course, it's absolutely another protein happens. But sometimes I just off top of this, frame shift mutations, they happened with a good intention, so with a good outcome. So very rarely, but sometimes in evolution we had examples when frame shift mutations so cause the formation of absolutely different proteins. So sequence is almost similar to another protein, but due to shifting of frame, so this protein is so exert so very specific function, and it's very rare, but it gives a evolution gaining there. Okay, then trinucleotide repeat mutations. So it's a special category. So you see, these mutations are due to repeating of trinucleotides. Okay, specific nucleotide sequence that undergoes amplification varies with different disorders. All affected sequences share the nucleotides guanine and cytosine. In example, fragile X syndrome, we will discuss it in next lecture tomorrow. Prototypical for this category of disorders, there are 200 to 4,000 tandem repeats of the sequence CGG within a gene called FMR1. So, repeats are happen there. So this feature discussed in greater detail later. Blah blah blah. Inheritance. They take part in inheritance. Okay, alterations in protein coding genes other than mutations. Alterations in protein coding gene other than mutations. So you see, not we don't only we don't only can change this gene. We can undergo other changes like amplifications, deletions, and translocations. Uh, So it amplification, it's increase of specific gene number, Deletions is absence of some gene number, so we delete, yeah, from word delete. And translocation, we just one gene is appeared in other area, in other chromosome, for example, in other place, and then our body can translate this gene, can transcript and then translate these genes, and that's why we can produce it. So pathogenic germline alterations involve contiguous portion of a chromosome rather than a single gene, such as in 22q microdeletion syndrome. It will be discussed later. Okay, then alteration in non-coding RNA. So you remember we have non-coding RNA. So in first chapter we discussed it, so you can imagine. So this non-coding RNAs, they take part um, in regulatory functions, and in alterations there, so they usually lead to, so if some important non-coding RNA is affected, so usually it leads to early death of embryo, so it's like miscarriages. That's why very rarely we can find it in a live person. Yeah, also, yeah, one thing I want to share to you, so which is hardly understandable by usual people, non-medical people. So we all genetic diseases we can see now, they are very mild diseases. These mild diseases because we so they are so mild that we can survive. So if you ever interested in embryology, so you see that how many genes are involved in proper development of the body, how many genes are taking part in formation of each organ, each part of, so each compartment of the body, in each bone of the body, yeah, so each little piece of the body is controlled by a lot of genes. And just imagine that one of this gene, so one of these most important genes is damaged. So what happens? So of course no development happens, and then development will be interrupted in very early period, even sometimes even before the implantation. And even if implantation happens, it wouldn't allow it to develop, and then it might lead to miscarriages, yeah. And sometimes what we name as um, so not miscarriage, I mean what we name as infertility, so actually there are miscarriages because fertilization happens, but it cannot get attached to the womb sometimes. So in some cases, that's why in half of so and actually in we have statistics that half of miscarriages, so 50% of miscarriages, and when we get analysis of these miscarriages, we can find some very extreme genetic lesions in children, in a child. So in 50%. So sometimes even chromosomal abnormalities. So that's why sometimes miscarriage is not a bad thing. So miscarriage is a way of our body to clear the womb from a genetically wasted person. Okay, but every disease we have, every disease we know. So it is a result. It results only because we can survive even without this gene. Yeah, we don't we feel very comfortable because of it, but still we survived. Okay. With this brief review of the nature of abnormalities that contribute to the pathogenesis of human diseases, we can turn our attention to the three major categories of genetic disorders. So now this you have to remember. So which genetic disorders we have to remember? Mendelian disorders. Mendelian disorders. So they are present in. You remember Mendelian rules in biology, yeah, and all genes which match to this definition, that abnormalities which shared by Mendelian disorders, so it's autosomal or autosomal recessive or autosomal dominant diseases which are present, which are shared by Mendelian rules. So they are in this category, of diseases. Okay, then we have complex disorders involving multiple genes as well as environmental influences. So we usually don't talk about this like genetic disease, but we don't talk about like disorder, yeah. But actually, it is anomaly. So we have every of us, every of you. So here you have some genetic lesion, so complex disorder lesion. So which is, you see, it's multiple genes and also environmental influences. So they can activate some of these genes. Some of them are not activated. So as example of this, I can say to you that, for example, cancer genes. These genes which are responsible for cancer development, they are complex genes. You remember, yeah, in lecture we discussed. It's usually complex genes. They have a lot of different genes which are responsible for, development of this, development of tumor, and also we have to have some mutations there, yeah, acquired mutation. But we have a lot of predisposition factors for the cancer. And one example, I give you example of myself. Example of myself. So I have a predisposition for lung cancer. Yeah. So my a lot of my relatives from father's side, they died because of lung cancer and respiratory tract cancers. And yeah, that's why. But all of them, all of those who had died from lung cancer, they had a bad habit, smoking. They smoked more than 20 cigarettes per day. And of course, if I want to avoid, and I have this complex, possibly I have, yeah, but these multiple genes. But if I don't want to die because of cancer, so I just don't need to smoke. So I just need to avoid smoking. That's it. That's why that's how this complex multiple genes are working, and that's how environmental influences work on these genes, because it's a very complex system, and we discussed in the first lectures. Okay, and then a third one is diseases arising from chromosomal abnormalities. So chromosomal abnormalities, all of you heard about Down syndrome. We have other syndromes, which we'll discuss in next lecture. But these abnormalities, chromosomal abnormalities, also have to be mentioned here. And also we have a category which is named as other genetic diseases. There might be involved single gene mutations, which do not follow simple Mendelian rules of inheritance. So they have non-classic inheritance patterns. So, for example, from triplet repeats mutation, from mutation of mitochondrial DNA, transmission of influenced by an epigenetic phenomenon. It's genomic imprinting. They will be discussed later. And then let's discuss the most well-known by you and well understandable by you. It's Mendelian disorders. Mendelian disorders. So it's when we have one gene responsible for one definite function for one protein, yeah, and it is shared in dominant or recessive way. Okay. So mutations involving single genes follow one of the three patterns of inheritance: autosomal dominant, autosomal recessive, or X-linked. We discussed what is autosomal. So you discussed it in biology, I guess, and in even in school, I guess. So a single gene mutation may have many phenotypic effects. So it's known as pleiotropy, because one gene is responsible for a lot of proteins in in different parts of the body. Because I remind you that every cell of our body contains similar amount of genes, and the same gene which is present, for example, in eye, and same gene which is present in, I don't know, in digestive tract, yeah. So it might be used, but it will be used in different way. So that's why we have pleiotropy. So one gene, if even one gene is changed, we have a pleiotropy of phenotypic effects. Okay. So mutations at several genetic loci may produce the same trait, genetic heterogeneity. Okay, so this best example is Marfan syndrome. It's results from basic defect in connective tissue, is associated with widespread effects involving the skeleton, eyes, and cardiovascular system. It's because of damaging of one protein. We'll discuss it later, and due to this, so we have a lot of effects. So one protein is damaged, but this same protein is used by um, different organs of our body, and that's why we still so we have different phenotypical changes. That's why we don't have gene, for example, of extra finger. Yeah, so we don't have gene of extra ear. I don't know why we don't have specific gene of some particular changes in our body. No, we have all the genes we have. They affect entire body because the same genetic composition, same DNA is present in every cell of the human body, and that's why don't surprise. It's not surprising for us that we have entire body changes um, in this case. Okay, then we'll discuss it. Also, recognition of genetic heterogeneity not only is important in genetic counseling, but also facilitates understanding of the pathogenesis of common disorders such as diabetes mellitus. So you see, diabetes mellitus. Okay, we'll discuss it in endocrine system. It is now increasingly recognized that the phenotypic manifestation of mutations affecting a known single gene are influenced by other genetic loci, which are called modifier genes. So later we'll discuss what is cystic fibrosis in next lecture, I guess. Modifier loci can affect the severity of extent of the disease. And okay, and and we don't forget about prenatal genetic screening, especially in high risk population. So Ashkenazi Jewish descents, they're very abundant in USA, which are very abundant in USA. They have a very increased risk of these genetic disorders because of what? Because Ashkenazi Jews they used to live in a very closed, closed communities, yeah, and very soon after a few generations, yeah, everyone is a relative to another or each other, yeah, and that's why a lot of diseases they can they have a recessive. Okay, we'll discuss what is recessive. Okay, we'll discuss this future. Okay, but they have a high risk of genetic disease. Okay, transmission patterns of single gen disorders. So this we have autosomal dominant inheritance. Autosomal dominant inheritance, we discussed it in biology, yeah, and you remember that autosomal dominant inheritance, it's when we have 22 chromosomes, and if we have gene in at least in one of this chromosome, it leads to some changes in genetic in phenotype. Uh, so these features, which features you have to keep in mind? With any autosomal dominant disorder, some patients do not have affected parents. It seems like counterintuitive. Yeah, so if we have autosomal dominant disorder in biology, they said it is always parents. So one of parents has this disorder, and one of parents share this disorder to this. But actually, no. Sometimes we have its gain of function mutations which happen during the before the fertilization or during the gametogenesis or during the fertilization. Some gain of function mutations might happen there, and that's why patients with autosomal dominant disorders they don't have a they don't affect as a they don't affect parents. So that's why it doesn't mean that if parents don't have the disease, so that this is autosomal recessive. No, it doesn't mean. So sometimes it's autosomal dominant. Okay.
Sir,
Yes.
sir, autosomal dominant will always, sir, phenotypically show, sir. If someone have autosomal dominant mutation, sir.
Absolutely right. Absolutely right. But I mean that parents not always share it
Yeah.
to children. So because might be it's just mutation directly of these children. So this child, yeah, or this offspring. So, no, but not of parents. So, and that's why sometimes parents don't have the disease, but children has in autosomal dominant way. And
Okay.
they,
And sir,
yes.
ratio of male and female will be same, sir, in autosomal disorders.
Yes, of course. Yes, of course. So we'll discuss. So of course now we have no difference between males and females in both of these ways. So males and females are so sharing it absolutely similar and with the same sequence. But point is that we have X-linked chromosomal X-link X chromosomal linked diseases, and we will discuss it. So after one point. Okay, then clinical features can be modified by reduced penetrance and variable expressivity. So, what's the point? We have homozygotes and heterozygotes, yeah, and in sometimes in heterozygotes this autosomal dominant mechanism so is little released because we have overlapping function of another gene, and that's why sometimes in some autosomal dominant disorders we have some phenotypical changes, but they are not so severe to be treated like disorder. So one of examples is in sickle cell anemia. Yeah. So heterozygotes of sickle cell anemia people, they don't have some clinical manifestations. They don't have sickle cells in their blood, but they are tolerant to malaria, and they don't suffer with malaria so hard. So that's an example of reduced penetrance and variable expressivity. In many conditions, the age at onset is delayed, and symptoms and signs do not appear until adulthood. So this is Huntington disease, for example. And autosomal dominant disorders, a 50% reduction in the normal gene product is associated with the clinical signs and symptoms. Okay, Okay, so this is hereditary. So how hereditary of this present? So it will be discussed. So you discussed in biology, so I don't want to stop on it. Yeah. Then disorders of autosomal recessive inheritance. So what is disorders of autosomal recessive inheritance? So usually we have to have two. So we have is working only in homozygotes, because usually we have two chromosomes. Yeah, in each chromosome we have some genes which overlap in function of another one. And so here I always give a example of it. And so just imagine you are in hospital, yeah, and your supervisor said to you, so bring me I don't know, urethral catheter. Yeah, so just bring me a urethral catheter. So he give you order, and then you come to go to the store. Yeah, so and then you see that so stock in the stock you see that in both on both letters you can see the catheter. You take from each of them, so you take one and then bring it to your supervisor. Yeah, just understand the situation. Yeah, but then next time, same supervisor said to you, bring me catheter. Then you go to the same stock, and you find out that in one letter it's absent, in other letter it's present. So you bring it from that part when it's present. But another part of stock is without these catheters. But you can take it from other side. Yeah. So and that's a principle of recessive inheritance. So if one gene is lesioned, so we can take it from another side. We can take it from another chromosome. We always have two chromosomes, so which can overlap another, one function overlap another function. Okay, but in case of just imagine the situation, the scary situation when your supervisor send you for something. Yeah, send you for a syringe. Yeah, for example, bring me syringe right now, very fast. So another way patient will die. And then you run out there and find out that there are no syringes in stock. And what would you do? So you would panic, and same panic happens in the body, in our body, when we have autosomal recessive inheritance, when two genes which you recessively inherited from your parents because of homozygotic, because you get homozygotic for recessive gene, so your body is in panic and can produce some very important protein, for example. Okay, when it happens, so usually autosomal recessive inheritance happen usually in case of relative marriages. Relative marriages. So because so just imagine. So every one of you. So as we discussed before, after every cell division, after every next population, next generation we produce a lot of lesion genes, and usually most of our genes, so most of genetic disorders we have, potentially we have, they're recessive, they're autosomal recessive disorders, and they are overlapped with another chromosome and what and just imagine, so which chances that next time, so your chromosome will encounter with another chromosome with the same lesion. There are myriads of genes. Yeah, we have myriads of myriads of genes which are working properly, and one lesion doesn't matter. Yeah, it seems like this, so it doesn't matter. So one little lesion, so will be floated around, so and will be dissolved in population. Yeah, and that's a desired feature. For our body. But when marriages happen in relatives, for example, your grandfather, okay, grand grandfather had one lesion, yeah, and then and the grandfather born one okay, one daughter and one son, yeah, and then outspring of this son, and so your grandfather and your wife, grandmother, for example, and then your step cousins, yeah, so your cousins, and you're far from them. Can you think, okay, so we are not relatives anymore, yeah, so we are far from each other, yeah, but uh, but one gene disorder which were shared by this grand grandfather to your grandfather and grandmother, they somehow we have chances to get joined in your child, get joined in your child. That's why it's prohibited in most of cultures. That's why relative marriages are prohibited in most of cultures, and sometimes in and also you heard about this, that in Hungary and in Iceland, they have programs by which they attract new settlers to their country with another genes, just to refresh the genes, because there are many relatives in small countries, and these relatives they get a collection of a lot of genetic lesions, and it's might be they don't produce some specific syndromes, yeah, but just in general, person gets weaker because a lot of these autosomal recessive genes are activated, and some proteins are not working properly. Um, okay, and this is and next let's talk about X-linked disorders. X-linked disorder. So you remember that in females we have two X chromosomes. In males we have only one X chromosome and Y chromosome. So a Y chromosome is an insufficient one. So Y-linked chromosome is a very small and and not effective. Y chromosome carries only SRY gene and also some genes which are responsible for hairs of ears. So very not important gene. So only one gene is important, SRY gene. So it's a sex determining gene. So it's responsible for protection of. So in embryology, we will discuss it a little bit in next semester, I guess. That SRY gene is a gene which protects embryo from influence of mother's hormones on the embryo, and because actually estrogen and progesterone, which present in the female's body during the pregnancy, it influences the female development of embryo. But if we have SRY gene, we are protected against this influence. So that's why that's the role of SRY gene. So which it prevents a female type of development. But so but if we don't have SRY gene, so of course all the development is controlled by estrogen and progesterone. Okay, then X-linked disorders. So X-linked disorders usually affect sons. Yeah, so also usually affect males only, because it's very rare condition when we have both X chromosome lesion, both X chromosomes. So it's homozygotic chromosome lesion. So it's possible. It's possible, but very rare, very rare. And usually. X-linked disorders are present only in males. Yeah. Why it's okay? Why we don't have dominant X-dominant lesions? Yeah. For example, why the for example, you remember that X chromosomes in females they randomly in each cell. So one X chromosome is shrinked, another X chromosome is remained. Yeah. And so one is shrinked, and for example, you see it in Bart's body in neutrophils. Yeah, this shrinked X chromosome is present like Bart's body there, and in each cell it goes randomly, and that's why approximately 50% activates this X chromosome. 50% of cells activates another X chromosome, and that's why generally, so that's usually enough to have the normal functioning of this each of this X chromosome. So only if in way of recessive disease, so this X chromosome, if in a homozygote disorder of this X-linked chromosomes, the X-linked disorder, it can cause pathology, but usually it's not. But in sons, we don't have another X chromosome which can overlap functions of this X chromosome. We have a Y chromosome which has no alleles similar to X chromosome, and that's why in males we have so an affected male not transmits the disorders to sons, but all daughters are carriers. So that's a male which has some disease, so already suffer with disease. They cannot transmit to sons because sons are carrying the Y chromosome, right? And but all daughters, he will share the gene. He will share this gene, so they will be carriers of this X chromosome with some disease. So which diseases are there, we'll discuss later. But I hope you heard about it. Okay, and then let's talk about diseases caused by mutations in genes encoding structural proteins. So now we'll start it today, and then we'll continue it tomorrow. So today we'll discuss one example. It's Marfan syndrome. All of you heard about Marfan syndrome. Yeah, all of you seen this Marfan syndrome. Uh, okay, no, might be you didn't seen it, but you haven't mentioned it, but you've seen Marfan syndrome. So the same main disease is caused by fibrillin 1 protein. This glycoprotein is it's a very important component of microfibrils. So uh, this secreted by fibroblasts, and in their important in microfibrils, they scaffolding the deposition of tropoelastin. And you remember, tropoelastin is a part of so it's before the elastin formation. Tropoelastin is a precursor of elastin formation, and fibrillin is taking part in fibrillin 1, so it takes part in a normal proliferation. So not okay, it's polycondensation of this fibrillin 1, and polycondensation of elastin, sorry, and normal deposition of elastin depends on fibrillin 1. But if fibrillin 1 is injured, so what happens? Of course, elastic fibers will be not properly deposed. So this which genes are FIBN1 gene? So that's this locus 15q21. So whatever it means. So don't, so just leave it. So no need. So a prevalence of Marfan syndrome is estimated to be 1 in 5,000. So it's, you see, it's very abundant disease. So it's for example, one student of International Medical University. Yeah, so they are they have a Marfan syndrome. Yeah, approximately 70% to 85% of cases are familial, and the rest are sporadic, arising from the de novo FEBN1 mutations in the germ cells of patients. So you see, main idea. Okay, pathogenesis. Okay, many of the abnormals of Marfan syndrome can be explained on the basis of structural failure of connective tissue. Some, such as overgrowth of bones and difficult to relate to simple loss of fibrillin, to relate to simple loss, it can be explained by loss of fibrillin. It is now evident that loss of microfibrils gives rise to abnormal and excessive activation of transforming growth factor beta. And you remember that was the function of transforming growth factor beta. So normal microfibrils sequester transforming growth factor beta, thereby controlling the bioavailability of the cytokine. Excessive transforming growth factor beta signaling has deleterious effects on vascular smooth muscle development and the integrity of extracellular matrix. So you see, TGF beta overproduced, and it causes a specific clinical manifestation of this disease. Okay, we have skeletal abnormalities. It is the most obvious feature of Marfan syndrome. So patients with Marfan syndrome, they are very long. They're very high. Sorry, very high. They have abnormally long legs, arms, and fingers. And this long fingers, they are named as arachnodactyly. So if you remember, arachnos means spider, yeah. And so fingers like spiders. So they have higher arched palate, hyperextensibility of joints. So might be you remember that guys who is very long, usually guys with specific features of the eyes, they can how to write, they can flex their finger towards the palm, yeah, so from opposite side, and that's and might be it is Marfan syndrome, so it's very common feature. They have ocular changes. So ocular changes, it's cause bilateral dislocation or subluxation of the lens secondary to weakness of its suspensory ligaments. So you see, suspensory ligament is very weak, so because of no elastic fibers properly developed there, and it causes a little opening of the eyes. So this eyes will be like little rolled out. Yeah. Then and but most sudden effect of this is so of course everything we discussed before, it's not so bad, yeah, so we think it's okay, but actually Marfan syndrome is present in cardiovascular system. The affection of cardiovascular system is very dangerous because our arteries, first of all aorta, and even heart, they have a lot of elastic fibers, and possession of these elastic fibers normally it facilitates the integrity of this aorta. But in case of Marfan syndrome, they won't be so elastic, and they won't keep their shape of the aorta, and it causes the dilation of aortas, which sometimes cause aneurysmal dilation. It causes aneurysmal changes, and also you remember that in the heart we have fibrous rings which are present around each valve. Yeah, and due to this mitral valve are so you remember this activity of valves. Yes, that valves should be closed, totally closed in this orifice. But just imagine that fibrous ring which presented in a heart so get extended, and valves which are present here, they are insufficient to be closed, and that leads to insufficiency of mitral valve. First of all, mitral valve insufficiency or mitral valve prolapse or regurgitation happens. So, and this is in symptomal complex known as floppy valve syndrome. Okay. Uh, And so, but these lesions which are discussed, they are not seen in all cases. Now, we have a lot of variations because some patients may exhibit predominantly cardiovascular lesion with a minimal skeletal and ocular changes. Okay, now let's show you some person who are suffering with the Marfan syndrome. So my very famous person you heard about, so who suffered, so who suffered or suffering with it. So 1 minute, where is it? Yeah, take a look. Abraham Lincoln. That's one of the most famous person with Marfan syndrome. And after the disease of Abraham Lincoln, so so one of USA presidents. So after we understood that Abraham Lincoln has this disease, it was a murmurs that rumors that might be this disease increase the ability of brain. They might be this disease cause a better brain development, and because a very smart person he was. Yeah, and usually we think that genetic disorders are usually affecting the brain. Yeah, but actually not in this disease. So a lot of prominent persons all over the world they have this Marfan syndrome. Take a look on the fingers. Yeah, so very long fingers present, eyes little rolled out, so it's not very well shown here, and very extremely high person. And so okay, ears are okay, doesn't matter. Actually, Julius Caesar. So we have no confirmation of it. Of course, we have no genetic analysis of Julius Caesar, but according to definition of his appearance, so we can say that Julius Caesar also had this Marfan syndrome because of accessibility. Yeah, and this is very famous person, Osama bin Laden. Uh, Sorry, I didn't share it to you. Why it's not shared? Now, yeah. So once again, sorry, so some back happens. So Abraham Lincoln, no, now you see, yeah. So Abraham Lincoln is here, so with the long fingers and very high according to their to his uh, citizens, and Julius Caesar by definition, and Osama bin Laden. So he's very high, unlike other Arabic people around, so Afghani people, yeah. So near them, he is a giant. He is very long, but also very thin. So in this, he has very extensible joints, head, very extensible joints, and that's and it's obvious that he had the by face expression, by this uh, small signs, we can conclude that it's also Marfan syndrome. So we can argue about his influence on all the world, but anyway, so we understand that he was a very smart person, yeah, and that's why so was so hated by others. Okay, then Bradford Cox, I don't know him. Might be you heard about him, but I don't know nothing about him. But that's best example as I want to show to you. Michael Phelps. Michael Phelps. This is a fish man. Yeah. So actually, he must be Aquaman. Yeah. In franchise. So Michael Phelps is. So you see, we used to think that he's very healthy because he can swim for long distances. Yeah. For a long time. And long distances, and he's a very strong person, and it's hard to imagine that he has disease. So actually he has, and actually a failure in elastic fibers formation, they help in the growth of muscles. And actually here is not in this collection, Arnold Schwarzenegger also has a so it's possibly he has this disease, or another disease, Egler disease. But anyway, those elastic fibers impairment, so it doesn't restrict growth of muscles. Yeah, because elastic fibers are present around the muscles and in perimysium and epimysium, we have these elastic fibers, and they just restrict the growth in some time, but they don't have this restriction, and that's why they're very well muscled males. Then John Tavener, so I don't know who is it. So according to bones which are found in Tutankhamun, so it's we can conclude that this mummy, this Tutankhamun mummy has had the how to, had the Marfan syndrome. So also I don't know, Jonathan Larson, some who is it? I don't care. Same this Vincent Schiavelli. So it's well known because of One Flew Over the Cuckoo's Nest. Might be you seen this movie, and also Niccolo Paganini. So was the most famous. So how this a violin player, yeah. So violin player, and that's these examples I said to you. Okay, and then let's move to other disease. Other disease, so it will be last disease we'll discuss today. It's Ehlers-Danlos syndrome. Yeah, sorry, no, we'll discuss familial hypercholesterolemia also. Ehlers-Danlos syndrome. So Ehlers-Danlos syndrome is a group of diseases characterized by defects in collagen synthesis or structure. So you remember that we have a lot of collagens in our bodies. There are almost 30 collagens. They're synthesized, and we have distinct genes which are responsible for collagen synthesis, and that's why. Clinical manifestation of Ehlers-Danlos syndromes, they're very different, but we have something in common. Tissues rich in collagen, such as skin, ligaments, and joints, frequently are affected in most variants of Ehlers-Danlos syndrome. And very typically, it's skin fragility. Skin is extraordinarily stretchable, extremely fragile, and vulnerable to trauma. So that's a main feature. So by which we determine them. So by which we can see these patients. Okay, once again, okay, I didn't share my screen. So remind me to share my screen. Sorry, because really sometimes I forgot to do this. In Ehlers-Danlos syndromes, we have so once again, it's an injury to collagen, and its skin is fragile. Skin is extraordinarily stretchable, extremely fragile, vulnerable to trauma, and also structural failure of organ or tissues. So because collagen deposition happens mm, not only in skin, but also it happens inside the organs, in stroma of organs, and that's why a normal supportion function of connective tissue so might be also failed. And we have deficient synthesis of type 3 collagen. So type 3 collagen, it's known as vascular Ehlers-Danlos syndrome, inherited as an autosomal dominant disorder, and is characterized by weakness of tissues rich in type 3 collagen, so like blood vessels, bowel wall, predisposing them to rupture. Then we have a deficiency of enzyme lysyl hydroxylase. Lysyl hydroxylase is this one which uses vitamin C. Vitamin C. So it it forms a cross links between collagen molecules, which makes it much harder. And usually these patients manifest with congenital scoliosis and ocular fragility. So this is how genes are taking part in formation of scoliosis. And deficient synthesis of type 5 collagen resulting from mutation. This is inherited autosomal dominant disorder and results in classical Ehlers-Danlos syndrome. So you see, it's fragile skin, fragile and extremely extensible skin. So then one more example. So now I'll show you the diagram. Where Handler. Ah, okay. Okay. No, well, I didn't. I won't show it to you. Okay, but you can check this disease in the Google. Okay, then let's talk about diseases caused by mutations in genes encoding receptor proteins or channels. So here we'll start only with familial hypercholesterolemia. So and only this disease I'll explain, and that's enough. Familial hypercholesterolemia is a receptor disease caused by loss of function mutations in the gene encoding the LDL receptors, which is involved in the transport and metabolism of cholesterol. So actually, we didn't discuss it yet, so it will be well in details. We'll discuss what is role of LDL in cardiovascular system, because LDL is a low density lipoproteins. These low density lipoproteins they are taking part in, they are taking part in formation of disease which is known as atherosclerosis. Okay, 2 minutes break. Okay, LDL is a low density lipoprotein. So this low density lipoprotein is so can penetrate the endothelial wall, and it carries the cholesterol. So this is what's the danger of. So you remember that cholesterol deposits and the endothelium of blood vessels, and that's very dangerous. Yeah, in formation of heart attacks and even strokes. Yeah, and just imagine we have a population of people which has hypercholesterolemia, familial hypercholesterolemia, so in which too many LDL are produced because receptors to LDL, they play a role of coordinator of LDL production. They have a negative feedback. So if there are too many LDL are produced, low density lipoproteins are produced, these receptors sense it, and due to sensing of this, this due to sense of this LDL, they give a signal to stop producing LDL. But if we have this, if we have lesion in this receptor, so of course we don't have feedback of LDL receptors, and this LDL will be extremely increased, and this will be the it will cause the lesion of blood vessels. So here we have metabolism, LDL receptors, and then endocytosis, which happens properly here, and the storage of cholesterol esters, how it happened here. So it is explained here, but we won't stuck our attention to this, because we will discuss it much deeper in topic of cardiovascular system and how this cholesterol takes part in it. Okay, that's enough for today. See you next time. If you have any questions, you can ask me right now. So you have to keep your attendance here. Don't forget. Don't leave. Any
Okay, sir.
questions?
Uh,
Okay, if no questions, goodbye. Thank you. You can leave.